First of all, I would like to say that I'm really happy to be here again. Last year I was on the track too, talking about image segmentation. Today, some different kind of research of mine. I'm representing the Silesian University Technology as a PhD candidate and the future processing as a software developer. And I would like to talk about mind reading. I will say something more later. But the agenda for today is how does the brain work? Of course, I don't know how the brain works, but I will show you some kind of basic knowledge. I will talk about brain reading devices, so how we can read this brain activity, and the state-of-the-art applications. And I will not fly off to the future too much. The state-of-the-art applications are impressive enough to show it on the presentation, not go into science fiction style. And I will show you some code from my research, my PhD research, and it will be in Python, and how to start to read minds in Python. So, in the last episode, I hope I have... I have internet. So I'm showing this to show you what is my background in image segmentation. This is the presentation on YouTube from a year ago. So I'm showing it to promote the video, to promote the work of my colleagues from K3D Jupyter. There is some application about hard segmentation using this visualization library. And of course, to show you some nudge of what I was doing last year, because I will be relating to this work of mine. So how does the brain work? This charming picture shows perfectly what neuroscience call the catch-22 of the brain. So if the brain were so simple that we could understand it, we will be so simple that we couldn't. And this is this catch, and we still don't know how the brain works. We are working on it. And by this picture, I would like to promote the Wait But Why blog post about Neuralink. I will be talking some about it later. And of course, we are on the long road to the knowing the brain. Like the Professor Jeff Lichtman from Harvard said, if all we need to know about the brain was a mile, we are just a few inches on this road. So what can you see here are the building blocks of the brain, the neurons. And the picture on the left is from the work of Santiago Ramón y Cajal. He's a Spanish neuroscientist, very well known. And these drawings are from 1905, so more than 100 years ago. And today, the state-of-the-art methods are fluorescent photography of neurons. So we can say we know how the neuron looks like. But the bigger question is how we can map all these neurons in our brain in the structure. So we know what is the neuron, but we need to find the connections between these neurons. This is video from National Geographic channel on YouTube. And what you can see on this video are the photographs of slice by slice, like physically slicing the fragments of the rat brain. And each photograph is a one slice of nanometer accuracy. And what we need to do is to segment these images using, for example, deep learning methods. It's the state of the art here. For example, these flat fill-in networks are used in the article to segment brains. And what we can do, we can map this on the 3D plane and map the whole brain. This is the ultimate goal, to map the whole brain and to know the structure of the whole brain. So if we know the structure of the whole brain, one more thing. We can use projects like Neo Brain Game Simulator. Authors said it is the biggest in the world project to map the connectome of the brain. So connectome, how the neurons are connected. And it is not released yet, but they say it will be like collective intelligence attempt to map the whole brain. And this is the ultimate goal, and some kind of game to help them map the brain. I should Started, okay. And if we know the mapping of the brain, we need to simulate the electrical activity of this brain, because the task of each neuron is to collect the electrical activity of other neurons and to pass them to the other neurons. This is the basic idea. If we know the connectome of the brain, all the connections and all the neurons activity, we can simulate the brain. What you can see here is the digicortex simulator, and you can see some activity on the back of the brain. It is the visual area. And here we have 16 million neurons and 4 billion connections, and it's still 5,000 times less than in the proper human brain. And you need to know that this simulation was generated not on like a standard PC, but computational cluster. So even if we know the whole brain and we have software for simulating the neurons, it is still a challenge to simulate it on some hardware. So we are, in terms of hardware, we are long way ago. Okay, so if you are familiar with the typical mathematical model of artificial neuron, you need to know that the biologically plausible model has a time domain information. So not only input and output, but the pattern of output, like what is the pattern of spikes. So this neuron activity spikes when it collects the activity from other neurons, and in some threshold it will spike his own electrical potential. And this is some called it like a third generation of artificial neural networks, because in the same number of neurons we can accumulate more information because of this time domain. Okay. And this is the simulation of how it can work. So each neuron has its own pace of spiking or the electric activity. And we have on the input some kind of, for example, video or a sequence of the data. And we can interpret the output spikes or the trains, spike trains, it's called spike trains. We can interpret it as an input to other classifier, or just interpret the time domain of this data. And there are pretty many of these brain simulators or spiking neural network simulators. Last time I checked this list on the top, there were more than 20 of these simulators. The ones marked with Python with the snake are Python-based or Python API. And these are the most interesting ones. This digicortex is one you have seen on the slide before. And the one that is particularly interesting is this Spawn. It's functional brain. It can solve some tasks, like fluid intelligence tasks, like IQ test or something like this, just by simulating how the brain works, and like biologically plausible simulation. And after 50 years and thousands of research papers and works, we know that this complicated structure of neurons is somehow structured in areas of our brains. In each brain, there are some areas that are connected to some or responsible for some task. I would like to focus on the visual association area, so the back of the brain. This is where the vision processes are happening. And I would like you to know the motor cortex. So on the middle of the head, there is the brain area for all the movements and the coordination of your movement. So if we know somehow how the brain works in the basics, we can measure this activity. And it will be a short introduction to brain reading devices. I will start from the triangle, good, fast, cheap. You know, when you are making a product, you can do it good and fast, but not cheap, or good and cheap, but not fast. Then there's something on the middle of this triangle doesn't exist. In case of brain reading, we have precise, and I mean by precise, I mean how precisely you can spot the signal source in the brain, like in what accuracy. how fast, so I mean temporal resolution, so how fast or in which frequency we can measure it. And by safe, I mean invasiveness of this, or if it's safe or it needs surgery actually. It's invasive or not invasive. So if we want to do it precise and safe, we have functional magnetic resonance imaging. It is completely safe. We can measure with millimeter accuracy the signal source of the brain. If we want to have it precise and fast, we have microarrays of electrodes injected directly into the brain, but it needs surgery, so it's not very safe for you. And the most popular one is the EEG. It's fast, safe, but it's very imprecise. It's impossible to tell where is the signal source. It's like trying to understand a person in a crowded restaurant, but through the wall of this restaurant, something like this. And this triangle here was just for a presentation, because there are at least two more dimensions in these devices. One is a price, and in terms of price, the fMRI is quite a big cost, so it's not very usual to have one in your house or in your scientific institution. So the EEG is the most common or popular method of brain reading. And I will be focused on this here, the emotive epoch, the low-cost devices, like in general. I will talk maybe some of the professional EEG. I will be doing research on my PhD. Okay, and if you want to have some more details, I can invite you on the Better Future Processing, Better Future site to read more on my blog post. So what do I mean by mind reading? The term mind reading isn't like perfectly specified in literature or scientific works. It's usually in quotation marks, like a science fiction thing, with the mind reading. So I'm not talking about the left picture, trying like telepathy or some kind of knowing the mind of another human, but I'm talking about all methods of computational neuroscience that can make sense of the brain signals. So that's what I mean by mind reading, not like Black Mirror, maybe Black Mirror future, but I will be not talking about Black Mirror, I will be talking about reality. So last year, Facebook, it is a picture from Facebook conference, F8, last year, and they are trying to create a device that is able to type in your computer or your mobile phone 100 words per minute. It is 5 times faster than regular typing on the keyboard. And actually, this mind typing isn't a new thing. It was shown by Facebook, but the first research in this area is from 1988. And it was based on some potential in your head that is changing when you are looking at the letter on the screen, the specific letter, and this letter is highlighted. When it is highlighted, there is a special potential, and you can interpret it as an input to the computer. But what Facebook shown is a Stanford University research, and they didn't use this potential that I said, but motor cortex, just using one electrode in the brain. I will show the video. They were able to inject this electrode to the paralyzed woman's brain and use the motor cortex to control the cursor on the screen or the pointer on the screen. And it was quite smoothly, it was a big success. They used it also in robot arms. Oh. So this is the biggest success of this team from Stanford University, to give the paralyzed woman the ability to drink using the robot arm. This is from 2011, I think. Nature video from YouTube. You can see it as well. And the newest thing is the prosthetic arm that can even feel the feeling of touch in your hand. So you can control it using fully your brain thoughts or your brain signals, and even feel there is someone touching it. Of course, it's after 6 months of rehabilitation to use this hand or to control it in a proper way. So the next thing I would like to talk about is the Neuralink, the newest company of Elon Musk. And the mission of this company is to create high bandwidth connection between brain and computer. And what is the ultimate goal? Is to change the communication speed of today, because Elon Musk said, if we cannot beat AI, just join it. So just connect with the computer and don't be beaten by the AI, just join the computer. So the typing, talking, or reading is too slow for Elon Musk. He wants all of this made in thinking time. So just with the very high bandwidth connection, we can connect with computers and other peoples in no time. Actually, there are some articles with brain-to-brain communication, like playing Tetris between internet. And what was this research about? Oh, here is the Pong. There are many of the applications. One person has an EEG on the head. The second person has TMS. It's like a brain stimulating machine by magnetic field. And the person, the receiver one with this TMS, was able to learn what is the meaning of this magnetic field in his brain, and to rotate or to play Pong in the way that the person is seen, that the sender person is seen. So there was this very popular interview from September where Elon Musk had some smoking weed event. And he said that, I think we have something interesting to announce in a few months that is at least at orders of magnitude better than anything else, probably better than anyone thinks is possible. It is about Neuralink. And it is before he smoked the weed, so it can be more true than not. So I'm still waiting. It's been like 2 months from this event. And other applications, we will see about Neuralink, but what are the other applications are lie detection, and there is evidence that in Guantanamo, prisoners were examined by fMRI lie detectors, and there are articles that are saying that fMRI experts in lie detection are 12 to 17% better than traditional polygraph experts. And there is also identifying musical pieces using just brain device, so we can tell which music you are listening to in the same time by just reading your mind. And the most interesting or mind-blowing application is reading visual information, so reading by your brain activity, reading what you are seeing or even dreaming. This is a very popular research subject in papers right now. So what I would like to show you is the first fMRI from Purdue University from USA. They are using fMRI on the people that are watching some clips, video clips. They are training neural network to interpret the fMRI maps or the 3D maps and reconstruct images from it. This is the result. It is from 2017, the first research. And the second one, this is the structure of this network they used to reconstruct the images from fMRI data. I will not talk about details here. If you're interested in, you can check the article. Actually, the CNN network was used as a brain simulator or brain model of the human to reconstruct and to classify images of what humans see. And this is the research that many didn't believe when it was released. It is reconstruction images from EEG. EEG data is mostly very noisy and not very informative. It's not precise, like I said. But researchers from Toronto was able to find the meaning, to reconstruct the faces of images. I will show you in the video too. And it was a very famous application, like for a few months. It is from the beginning of this year. So just using EEG, like a professional EEG, like you saw, like for the $15,000, they trained the SVM, not deep learning. It's not deep learning. It's SVM to make meaning from brain signals and reconstruct the faces. If it is not faces from training set or the same faces, but just the whole face space. So we can see the application, how it works, the demo. And each face presentation, the brain changes the activity, and we can interpret it by this SVM model to reconstruct pixel by pixel, actually, the face we are seeing. And at the end, I would like to show you a little demo based on my article, the latest article, about using this low-cost device. I will show on the other slide. It is something between this NeuroSky, the one electrode devices, and the professional devices. It has 14 electrodes. It is like very simple to wear. Of course, we need some saline to wet the sensors because it is water-based, not like a gel or something like this. And it is quite cheap. Now we have Black Friday, so we can buy it for $640. If someone is interested, of course, you can see it after presentation, this device. I will not show the demo because the demos from the producent are actually based on EMG, so the activity of the muscles, not the activity of the brain. If you would like to use it like real mind reading, I will show you how. Okay, so what was this research about? This my research. We have faces of happy, neutral or happy and angry people. And we tried to find the difference in the brain activity for neutral faces and emotional faces. So there was the point of this research. We recorded and designed this experiment in OpenVibe. It is open source presentation software from INRIA Institute. And this small rectangle here was used for synchronizing this device with exact presentation moment. On one keynote, we heard that it's impossible to really find some exact moment in the video where some emotion can be noticed. But here we have just the presentation of a face by face, like a blank screen and face. And this smaller rectangle here is for synchronizing through the diode that I made. So what can we do to start in Python to process this information in Python? We have 14 channels, 14 time sequences. So we can use MNE. It is equivalent of MATLAB EEG Lab. It is the most popular one in scientific community. But we can use it in Python by just installing MNE, pip install MNE. And it's funded by some grants, scientific grants, American and French grants. And it is compatible with NumPy, SciPy, scikit-learn. And I will show you some bits of code in MNE. So first, I will load the data from this emotive epoch, from this experiment. It is synchronized, so it is called processed, because what we need to do is to synchronize the markers from this rectangle, these signals from the diode, with the real markers, because they are changing or they are not synchronizing well in this device. So I will run this to show you how this signal looks like. So this is the signal from these 14 channels, or actually 12 here, because 2 electrodes are used for this diode. They are like connectors for this diode to synchronize the events directly in the signal. This is completely messy and noisy. I was very skeptic about this at the beginning of my research. But it shows up that after some processing, here I know that the bad channels are T7 and F3. I will not talk about detail what are the meaning of these letters and numbers, but there are some standard systems of position in these devices on the head, and they are called by letter and number. And the preprocessing, we have setting the average of the signals. The set reference average, it is doing, it is just taking the average of all signals and re-referencing the signal of all channels to this one baseline. And of course, we have filtering of the signal, so we don't want to have DC level. We want to have 0 level, so no just floating like in a 10 minutes range of time. We don't want these floats, and we don't want these very fast oscillations. So we are filtering the data. Now it looks better, this signal. And what we can do at the end, we can just select the epochs. And by epoch, I mean one phase presentation. So each phase presentation is one epoch. So epoch by epoch by epoch by epoch. And it was like 240 times in each experiment. Why we need this? Because only 5% of the activity we are measuring is connected to the task. So to get rid of any different signals, we need to average like few or several dozens of this kind of epochs. We can find some similarities between these epochs, like this peak in the beginning of this epoch, but only after averaging we can notice. Oh, we cannot. Oh, okay. This is the average signal of all epochs and all channels. And we can see at the time of 170 milliseconds, the peak, the down peak, and it is connected with phase processing of the brain. It is well-known potential. It is called event-related potential of phases, negative 170. And then we have potential connected with emotional processing. It is all like found in the psychological research, connected with emotional processing. And we have channels here. The channels here are from the back of the head, the occipital side. And we can see on the topograph map here that the blue back of the head means low or negative potential, and the red forehead means positive potential on the front of the head. And what we can do using this diode synchronization, if you use just this device, what I would like to talk, is that it's not very precise in terms of time synchronization. So if you want to use it in like mind-reading experiments, you need to be aware of these synchronization problems. You can analyze the probability signal probability density of frequency. I mean, just use Fourier transformation and analyze the frequencies in the signal. And you can also make a classifier using these frequencies. But to use time domain or just event-related potentials, you need this modification. And at the end of this research, we were able to statistically, of course, because we can average these epochs and measure only statistically if it's possible to distinguish between neutral phase and emotional phase. So this is the result for one participant. So for neutral phases, the positive signal in N20 250 was different than for emotional ones. And it was statistically significant, but still it is a very cheap device, and the only goal was to check if there is any difference. And there was a difference, it was published, and now we are going further and trying to find more applications of these low-cost devices. Of course, I'm also working now on research-grade devices and trying to make some processing because I'm a computer scientist and trying to do some machine learning on it. In terms of machine learning, we have more different libraries in Python. Here you've got slide, if you want to use some of them. Especially interesting is this one, NI Learn. It is based on scikit-learn, but more prepared for EEG processing or brain signal processing. And thank you. You can contact me on these different channels of communications. I hope you enjoyed.